Hi everyone, I'm Dora and this is the biology video for the topic viruses. So we can start with the structural features common to viruses. They have a small size between 20 and 300 nanometers in diameter. They have a fixed size because they do not grow. They use nucleic acid as genetic material, meaning that they have genes made of DNA or RNA, and they use the universal genetic code. Viruses have capsids made of protein. So before they're released from their host cell, their genetic material is enclosed in a protein coat called the capsid. And this is made of repeating protein subunits. Viruses released from host cells have no cytoplasm and contain no or very few enzymes. The diversity of structure in viruses. So they're very diverse in shape and structure. We can first mention the diversity of genetic material. Positive sense RNA viruses use their genes directly as messenger RNA. Negative sense RNA viruses transcribe their genes to make messenger RNA. And retroviruses make double-stranded DNA copies of their RNA genes and then transcribe the negative sense strand on the DNA to produce mRNA. So they have very different ways and techniques to produce mRNA, which creates a diversity in the genetic material. And then we have enveloped and non-enveloped viruses. To be released from their host cell, viruses bursted in a process called lysis. So some viruses become covered in a membrane during this process. This is particularly common in viruses that infect animal cells. The phospholipids in the membrane around the virus are derived from the plasma membrane of the host cell. So the proteins, mostly glycoproteins, come from the virus itself. And the membrane helps the enveloped virus to make contact with a host cell and infect it. But other viruses do not become enclosed in a membrane, which are non-enveloped viruses. Most viruses that infect bacteria or plant cells are non-enveloped. Now we have the lytic cycle of a virus. We have the lysogenic and lytic cycles. In lysogenic cycle, the viral DNA becomes integrated into the bacterial DNA molecule, so new whole virus particles are not produced. In the lytic cycle, the virus reproduces and then bursts out of the host cell, killing the host cell. This is a table for 3 different types of viruses. In comparison. So, for example, the bacteriophage lambda is a DNA virus that uses either a bacterium or an archaean as its host. It's a non-enveloped virus, and the genetic material is one double-stranded DNA molecule. The distinctive features of bacteriophage lambda is that it can follow either a lytic cycle in which it reproduces and then kills the host cell as it bursts out, or a lysogenic cycle in which it integrates its DNA and does not kill the host. And this is a picture of the virus. And then we have the infamous coronavirus. It's an RNA virus with a crown-like shape that uses an animal cell as its host. This is the crown shape. It's enveloped, so it has a membrane derived from the host cell. It has one single-stranded positive sense RNA molecule, and the pandemic of COVID-19 is an example of zoonosis because it was passed to humans from another species, probably a bat. And then we have the HIV virus. It's a retrovirus, which is a virus that converts its RNA genome to DNA after infecting a host with reverse transcriptase. It is also an enveloped virus, and it has 2 copies of a single-stranded RNA molecule. It contains the enzyme reverse transcriptase, which makes a double-stranded DNA copy of the viral RNA genome. This is then integrated into a host cell chromosome. The host cells for the HIV virus, which affects the immune system, are T helper cells in the human immune system. This is the demonstration of the Leidig cycle. It has the steps and descriptions you can see here. In Leidig cycle, as I've said, after infecting the host cell, the virus makes the cell burst out and kill the cell. And the viruses can also spread to infect other host cells after killing the one. And then we have the lysogenic cycle's demonstration here. We have the steps, integration, and cell division. So it does not kill the cell, unlike the Leidig cycle. While a virus remains in the lysogenic cycle, it's temperate, so it does not kill its host, and it causes minimal harm. But temperate viruses can benefit the host cell because their DNA may include genes transferred from a previous host, which increases genetic diversity, facilitating evolution. The evidence for several origins of viruses from other organisms. We have 2 different types of hypotheses. One is progressive and one is regressive. Scientists think that the regressive hypotheses are more likely to be accurate. So the progressive hypotheses mainly include the idea that viruses were built up in a series of steps by taking and modifying cell components. And the regressive hypotheses support the idea that viruses developed from cells in a series of steps by loss of cell components. This fits with the observation that both viruses and bacteria show variation in complexity and self-reliance. Rapid evolution in viruses. So, viruses can show extremely rapid rates of evolution. Even during an infection of one person, a virus can undergo heritable changes. It can evolve. So the 3 main reasons for this rapidity is that evolutionary change can only happen between one generation and the next, so it's limited by generation time. In humans, the average generation time is about 25 years, but in viruses, it can be less than an hour. Additionally, evolution depends on genetic variation. So the ultimate source of this variation is mutations, and the mutation rates tend to be high in viruses. This is particularly true in RNA viruses, such as coronaviruses, which do not perform any checks or correct errors made during replication of their genetic material. Then we have the 3rd reason, which is evolution being the result of natural selection acting on variation in a population. So the intensity of natural selection on viruses is high, and because of that, they tend to evolve more rapidly. 2 examples of rapid evolution are the influenza virus and the HIV virus. So we already mentioned the HIV virus on the table above. Because of these reasons, these 2 examples tend to evolve very, very rapidly. This is the end of the viruses topic, and thank you so much for watching this video. I hope it was helpful.